kind of nice to escape and do somewhere else. Have your own hotel room and not have anybody be able to get you immediately. That kind of thing. Yeah. Hi, Derek. Did you get my email?
I did. Thank you so much.
I'm sorry about the delay.
Oh, no worries. You know, the thing is, I was able to do research on my own, and like that was probably like the one book that stopped me question. I was like, I ordered everything else around it, so.
Oh, cool.
I'm in good shape.
Cool. Okay. Great. It sounds like it's going to be terrific.
Hi, Jen. How you doing?
Good. How are you doing?
I'm doing well. Are you going to be at the party today?
I am. I'm going to be delivering the food.
Oh, excellent.
Thank you for doing that.
Okay, good. But I'll be with him.
Sorry, I'm missing.
And the dog and... Well, fucking focus. Yes. Exactly. As long as it's a given.
Oh, actually, I'm taking one class at the it's on.
Good afternoon, everyone. Good afternoon, and thanks for coming out on a fine fall day. I know we will make it worth your while. Welcome to The Boundaries of the Human in the Age of the Life Sciences. This is a 2-year Andrew W. Mellon-funded project of the Institute for the Arts and Humanities exploring the boundaries of the human in the age of the life sciences. This is our second of 9 visits over the course of the next 3 semesters. The project is then culminated in the spring of 2017, where all 9 participants come back and we workshop what we have learned over the last 2 years and take it from there. So mark your calendars now. April 2017. Don't really have a date yet. Our guest today is Hannah Landecker. She earned her PhD in Science and Technology Studies at MIT and her bachelor's in Cell and Developmental Biology from the University of British Columbia. She has taught and researched in the fields of history of science, anthropology, and sociology. At UCLA, she is cross-appointed between the Department of Sociology and the Institute for Society and Genetics. Her first book, Culturing Life, How Cells Became Technologies, received the Suzanne J. Levinson Book Award from the History of Science Society. She is currently working on a book called American Metabolism, which looks at transformations to the metabolic sciences wrought by the rise of epigenetics, microbiomics, and cell signaling and hormone biology. Professor Landecker's work has fascinating implications for how we understand biology, but perhaps even more fascinating implications for how we understand things that have long been considered topics other than biology, such as history, philosophy, temporality, and disability. Professor Landecker's talk this afternoon, The Biology of History, From the Body as Machine to the Metabolic Community, will give us some sense for intellectual breadth, of course, but I think it will also do more than that. It will give us some sense of how her work allows us to think anew in the social sciences and the humanities about, literally, the meaning of life. Please join me in welcoming Professor Hannah Landecker.
So thank you very much for the invitation to come on this fine fall day and share with you some of my thoughts about the intersections of various disciplines, the boundaries between them, and crossing the boundaries between them. And I'm going to be talking from the project that Michael just mentioned called American Metabolism. So I thought I'd situate you first in where the project is coming from and then dive into some deep detail, first about the body as machine and then about the metabolic community. Can everyone hear me all right? Great. Okay. So here's a little bit of a insight into the structure of the larger project, just so that you can maybe see how the extract that follows fits into the larger thing. It's a not very ambitious scope of approximately 200 years, where the first section of the book really looks at the concept of metabolism as a product of the industrial age. And the logic of conversion, which will become very significant for us here, is simply the way that we came to understand eaters as converters. And things that are outside of the body, once they become ingested, become totally converted into that body. That's not an inevitable idea. And so knowing how it came to be is very important also for understanding the constitution of biological ideas and, of course, also social ideas of individuality or individuation between bodies and environments. The second part of the book, which I won't address much today, and in fact I'm going to skate over the 20th century in about 30 seconds, so maybe we can take it up in question period, is about the enactment of these logics of industrial metabolism in the world. So one of the things that I like to say is that these ideas were not just floating above the landscape in some way and changing up there in the sky, but really finding material instantiation in the way that food was produced, animals, bacteria, and plants were manipulated very actively. And then the third part of the book is really based in the present and an argument about the rise of a post-industrial metabolism. I also wanted to situate you in methods because this is a very interdisciplinary symposium. I'm very pleased to be part of such a sequence because at UCLA I live literally on two sides of campus. I get a lot of exercise going between the Division of Life Sciences and the Division of Social Sciences. So I practice ethnography for the contemporary parts of the book, that is extended hanging out in the spaces of metabolic sciences. I have a lot of experience and training as a historian of science, but I also have a very literary bent. And in fact, at the University of British Columbia, I did as many courses in English as I did in biology, but UBC wouldn't grant me a double degree because they said there was no useful complementarity between biology and English, which of course just encouraged me to prove them wrong for the rest of my career. So I like to use ethnography, observation of contemporary sciences, to ask really different questions that we have asked before about the past of those sciences, and also use the deep historical work to map the present differently. I also like to read scientific literature as literature for its temporal forms, for its account of the human condition, or these days, the human condition is increasingly understood as the animal, bacterial, cellular, molecular condition. To give away the ending before even beginning the story, this is the sort of arc of my observations, is that we have gone from an industrial concept concerned with energy and manufacturing and chemical substrates, waste accumulation and labor and fatigue, to a biomedicine centered on concerns about information, or perhaps disinformation is a good way to put it, to regulation and dysregulation, signals, asynchronous signals, functional asynchrony, and sleeping and aging. One of my favorite events at UCLA lately was the renaming of the Center on Aging as the Center for Longevity. Which I think was a very hopeful renaming, that more philanthropic input would come in if it was about longevity. So one of the arguments of the book that's also central to my argument today is that what I'm presenting here by laying out this arc is not just a standard history of biology. We understand what a history of biology is, right? That we first had this concept, and people in laboratories did experiments with these kinds of instruments, and then, ideas changed, cultural context changed, and science changed along with it. That, of course, is the kind of work that I do as a historian of biology, but because perhaps of my earlier training as a cell biologist, I've also really come to appreciate the fact that biology itself is changing, that the cells, the bacteria, the bodies of 1900 are not those of 2000, and that the sciences that we have, let's say, of antibiotics for controlling disease, cause antibiotic resistance in the bacteria and the bodies of living things, And then at the center of microbiology, you find antibiotic resistance. So it's a kind of recursive structure where previous knowledge creates a landscape in biological matter that itself is the ground for further knowledge. So we can talk as much about a biology of history as we can a history of biology. A few other arguments that are really central to the kind of more detailed stories I'm going to tell you is that another thing that I really noticed, even though I've been doing this for many, many years, but I never noticed before, is that the history of metabolic science and thinking really reveals a long and concerted effort that has gone into separating metabolic things from reproductive things, or we might say metabolic things from replicative things in the genetic era. And so I always knew, but sort of just took for granted, that genetics and the biochemistry of intermediary metabolism were two separate disciplines, because they were obviously two separate things. But I've really come to appreciate that too is not given, not inevitable. And it is through the ethnographic work, looking at what people are doing, especially in epigenetics, that I've come to see and experience what Liz Wilson would call strange biologies that really fracture these categories of the genetic or the reproductive and the metabolic in really interesting ways. And so this sort of sense that I have of contemporary biology as a theoretical resource for rethinking all kinds of things that are fundamental to much more than biological science comes from this relationship there. Okay, so because we were talking about boundaries in the title of this seminar, I thought I would bring in Liz Wilson's work as a kind of touchstone in the theoretical resources that I myself am mobilizing here. And here she's talking specifically about contemporary feminist theory, but I think you could, in some ways, just put humanities theory or cultural criticism here and still have the structure be the same. She writes in a surprising number of contemporary feminist and queer texts, one of the core conceptual commitments is a repudiation of biological explanation. An anti-biological gesture is often the ignition that starts the theoretical engine. And of course, this is understandable in a rejection of essentialism, refusing to see biology as the ground for culture and society. In my own discipline of sociology, for example, you see this pushback and a claim for the fundamental causes of health disparities, for example, being social in nature, to push back against, let's say, genetic explanations. So we can understand this separation, but we don't necessarily now have to accept it. She further writes, it's my concern that we've come to be astute about the body while being ignorant about anatomy, and that feminism's relations to biological data have tended to be skeptical or indifferent rather than speculative, engaged, fascinated, surprised, enthusiastic, amused, or astonished. So I invite you to be enthused, astonished, surprised, skeptical, any of those things about the kinds of ontological crackings, let's say, that we are finding in today's strange biologies in these kinds of categories. Okay, so that was the introduction. I'm going to take you back now to the 19th century, and hopefully I'll get back out of the 19th century, because one of my experiences is that once you get into the 19th century, it's really hard to go anywhere else. And really, before I started this project, I had completely defined myself as studying biotechnology from 1900 to now. It was a very, intentional maneuver. But since metabolism did not exist before 1839, I had to at least go back to 1839. So I bet you didn't know that metabolism did not exist before 1839. And here's the man who coined the term, and he actually didn't coin it as a noun, not as a thing. Metabolism was not a thing, but it was a power, and it was a power of cells. So Theodor Schwann is very well known for this book, which here's the title for you, translated from the German, microscopical researches into the accordance in the structure and growth of animals and plants. So he's very well known for this book because it's the first articulation of cell theory. That is, that all living things are made of cells, that there's a unity between plants and animals because they all have cells, and of course a unity with the smaller single-celled beings that could then be seen through the microscope. Schwann was very insistent that life in all of these forms could be explained without recourse to spirit or mind or any vital force. And so for him, this was an argument about the chemical and physical powers of living things, and the cell was the agent there. So we tend to read backwards on Schwann a structural argument that all things are made of cells, but we should actually read back a kind of process argument that cells had the power to both generate new cells and to transform things chemically. And at the point where he is coining the term metabolic, he writes about those phenomena which result from chemical change, either in the cell itself or in the surrounding fluid. These may be called metabolic phenomena, and he actually writes in the Greek origins of the term, signaling that he's finding a new word for a phenomenon which had no word for it before, implying that which is liable to occasion or suffer change. And he also, for our purposes, very significantly notes that the metabolic powers, that is, the power of the cell to transform substances from one form to another, can be separated from those phenomena attending the formation of cells, which he called the plastic powers of cells. Now, why do I call this an industrial concept? He writes, the attractive and metabolic powers of the cells are set free only by a certain combination of molecules, as, for instance, electricity is set free by a combination of a zinc and copper plate. Now, he's not making a sort of loose metaphor here. He's thinking about Galvani's battery, where the mere juxtaposition of two kinds of metal in an acid bath could generate another form of interaction. That chemical change in the metals, together with the acids, caused current to flow. And as investigators of the time then noticed, current flowing through a wire also caused magnetism. So it was a time of thinking about conversions, right? How juxtapositions of matter could result in the generation of another form of interaction or a motive power of electricity, and in turn could be converted into magnetism. I would argue that it also wasn't a metaphor. It was not a metaphor that the cell was like a battery, because many of the early batteries were themselves formed of living things. Here are some depictions of the early frog batteries. Galvani didn't just make things with metal, he made things with parts that were composed often of frogs' legs. And so the notion of animal electricity and animal heat was very current in this time. Now, I also want to tell you that these things were not just esoteric matters of the laboratory, but indeed there were many aspects of the laboratory in that time that were themselves the product of an industrial era. So this is a little earlier than the 19th century, actually much earlier. Here's the cover of a text from 1681 from Denis Papin about a new digester or engine for softening bones. So this thing that you see here on the, where's the pointer? This thing that you see here, the picture that you see on the screen there, is a steam digester. Does anyone know what we would currently call that? A pressure cooker. Yes. And So we all know what a pressure cooker is, but we don't generally know that the precursor to the steam engine was the pressure cooker. And Denis Papin was very involved in the kind of inventive theory and tinkering that led to the steam engine. The significance for animal chemistry, or the investigation of the interconversions of matter, was that you could put stuff in here and hydrolyze it. You could split it apart under the force of heat and water. And so it was just one tool among many being pulled in from the industrial processes of the world at that time to convert animal and vegetable matter into what was hoped to be the principles underlying that matter. So a lot of work during that time was dedicated to trying to explain what food was composed of, what animal flesh was composed of, what vegetable matter was composed of, and how one was converted to another, but also how one could intentionally convert one to the other. So there's a very wonderful book by Emma Sparry called Feeding France, in which she argues that it was the industrial chemistry of trying to figure out the composition of foodstuffs and alcohols and soaps and candles and all of these important, increasingly mobile commodities that was the basis of chemical revolution in this time and changes the material culture much more than, say, the work of Lavoisier in discovering the element of oxygen, which usually gets all of the press in the history of chemistry. So this is another way in which metabolism was a fundamentally industrial concept, thought in and through both the use of industrial machines and for the making of new kinds of foods to circulate, the question also of increasing number of people housed in institutions. And so the pressure cooker allowed the making of gelatin from bones, for example. And the question was, could you use this economically useless stuff, bones, to make food, especially for institutionalized people, whether they were sailors on boats or the poor in poorhouses? And this was such an important social question that France's best scientists were brought to ponder the matter. So here is Claude Bernard, one of the 19th century's best-known experimental physiologists, who first came to consider questions of nutrition. He didn't use the word metabolism, probably very intentionally, through doing feeding experiments with gelatin to dogs, trying to figure out if dogs could live on gelatin. The results were ambiguous, and I would say also that the results were very ambiguous from all the aforesaid institutions because people didn't like gelatin very much, and it went off very quickly, and it didn't seem to hold up to its promise of being a very economical and nourishing food. However, Bernard did go on to work for the rest of his career around questions of what was happening on the inside of the animal body in nutrition. Here's a picture of him in his laboratory surrounded by debating individuals. And I could spend all of my talk on him, but I'll just tell you that his concept of nutrition was very central to what would later be called metabolism. He felt, like Schwann, that he needed to designate a separate time and space from words that had come before. He said nutrition and digestion are completely separate. So between digestion and excretion, this process of nutrition happened. And at the same time, Justus von Liebig in Germany is using the term Stoffwechsel. So we have Schwann's metabolic powers, we have Claude Bernard's nutrition, we have Liebig's Stoffwechsel, all trying to reach into this chemical interiority through which the dog does not get fat on mutton fat it makes dog fat. And here's an elaboration of Bernard's concept. One does not live by his present food, but by that which he has eaten previously modified. The food first disappears as a definite chemical material, and it's only after extensive organic work, after a complex vital elaboration, that the food comes to constitute the reserves, always identical, that serve for the nutrition of the organism. This is the explanation of the milieu intérieur, the mode by which the animal, the higher animal, is free, moves through a changing environment, ingesting all kinds of other organisms, but always remaining itself. And so this is a concept of physiology, but it is also a concept of individuality, of a constant way of making a boundary between inside and outside. The historians of science, Hans-Jörg Reinberger and Stefan Müller-Willi, have argued in A Cultural History of Heredity that one sees in the late 19th century a transition from heredity as a space of thought and practice to heredity as embodied in particular objects, the move from epistemic space to epistemic objects. So in this case, the model organism, the pea plant, etc. I would argue that equally, this transition of metabolism as a space of thought that's differently demarcated and named by these different characters that I've said to you. Also, at this point, there's a separation between heredity and metabolism that gets further entrenched by being embodied in different objects. And for metabolism, it really was the enzyme-catalyzed reaction that could explain this conversion of mutton fat into dog fat, or the conversion of food in its complex form to its so-called building blocks, a term which comes into currency at the beginning of the 20th century. So the story that I want to tell here about the 19th century is that you have this logic of total chemical conversion that happens on the inside of the body. It comes along with a politics of hunger, labor, and energy. I'm giving you a very abbreviated story here, just because I want to make it out of the 19th century, but I could go on at great length about the way in which these investigations were through and through motivated by the worry about the food and labor power of nations, the labor power of farm animals, the worry about trying to make farm animals grow to be bigger, on this problematic feature of them that you could feed them more and more and they didn't necessarily grow past their adult weight. So this politics of hunger, labor, and energy gets inserted into the cell through a revival of Schwann's cell theory at the end of the 19th century, and called metabolism, which is perceived to be a separate chemical power and process from the hereditary. It's only in the 20th century that we get the rise of the science of intermediary metabolism, which captures this formerly diffuse set of terms of nutrition and Stoffwechsel. And here we have J.B.S. Haldane in the biochemistry of the individual stating the ultimate aim of biochemists may be stated as a complete account of intermediary metabolism, that is to say, of the transformations undergone by matter in passing through organisms. What transformations were these? Well, this is the kind of thing that they were talking about, the transformations that things go through in these metabolic cycles, where at each state there is an enzyme-mediated reaction that turns succinate into fumarate, into malate, etc., etc. So this is Hans Krebs standing in front of a depiction of the Krebs cycle. So as I said, I'm going to really rush you through the 20th century and just sum it up like this. And these are actually two pictures of time, right? This is the time of homeostasis. This is the organism constantly changing in order to remain itself. These cycles just go round and round and round and round. They, of course, need input, but you find many, many formulations of the wonder of intermediary metabolism as the organism constantly in turnover in order to remain exactly what it is. So this is the picture of homeostasis and the keeping away of negative entropy. This, by contrast, is, of course, the temporality of intergenerational transfer and the sciences of heredity. Okay. So now I'm going to move you quickly into the present. There's a whole part of the argument there about how the ways in which those logics of metabolism were enacted in the landscape changed all kinds of food production, changed all kinds of production of antibiotics and all kinds of other things, to the point where you get a lot of changes in the bodies of the people, let's say, who are coming into the clinic, but also in the bodies of the organisms on which science is being performed. So, as many of you know, and as is countlessly recounted in the talks that I go to, there is a crisis in metabolic disorder in the world. This has, in turn, created an incredible flourishing in what became a very static field, that is the biochemistry of metabolism and its links with things like cancer and diabetes. It really began to take off again around the year 2000. So I've been observing this field, a very wide and kind of eclectic observation of all kinds of things that go under the banner of metabolism since about 2005. And I've really been struck by the transformations in the answer to the question of what is food. In the previous era that I showed you, it was a substrate. It was something that came into a reaction and was torn apart into pieces and then synthesized again to make something else. Here, however, in the contemporary, more often than not, I see food as a form of information or exposure being discussed as something that tells the body something, or as a signal that is transduced onto the inside of the body. And perhaps specifically to our interests today, in nutritional epigenetics, as something that leaves an imprint on the body, and specifically on the body of chromatin, that is something I'll explain to you, especially at certain periods of the lifespan, and persists in a way that is long after ingestion. So it's a different temporality than the homeostatic cycle, and it's a different kind of way of thinking about conversion, not totally converted anymore. So a few words about chromatin. Chromatin, by definition, is a colored stuff. It was named so because of experiments in the late 19th century with dyes, and the chromatin would take up dye, so it's called colored stuff, which doesn't tell you much about what it does. It is, by definition, both DNA and the proteins that DNA is wound around. So here you have this depiction of the 2 meters of DNA in any one cell that have to be squashed down into 10 micrometers in the nucleus. So this kind of folding of 10,000 compaction of 10,000-fold happens by winding the DNA strand around these core of 8 histone protein molecules and further compacting that down to make the iconic chromosome here. Of course, chromatin is not always in chromosomes in cells. For much of the cell cycle, it's slightly unwound, not in its chromosome form. The chromatin for a long time was seen as a kind of inert scaffold for just, holding up the precious DNA molecule in the right shape. The end of the 20th century, people began to think about this as being a much more dynamic structure. And thinking about the fact that when a gene is being expressed, the chromatin shape and form, its conformation changes. It opens to allow gene transcription. In those places that are silenced, such as in the ends of the chromosomes, the telomeres, the chromatin is very, very tightly compacted and silenced. And so this role of shape, of topography, or topochemistry, we might say, in the mediation of what a genome was in the world, not just the sequence underlying it, but how it became manifested as transcription and translation, began to be a real focus for research. And very, very interestingly, you see this kind of formulation as chromatin as the body of the genome, the physiological form of our genome, or as Eva Blanca put it, the gene's phenotype. Okay, so in some ways, I've been doing an ethnography of chromatin, following it around in laboratories. Here's an interesting depiction of the relationship between food and chromatin, or food and DNA. It's very mechanistic, but it still, I think, speaks to the general logic here. Here you have the pizza. I think that's a pizza. Is it pasta? Pasta with sauce, yes, okay. Being sort of taken apart, right? And so we do have this logic of conversion still active here by this machine, and it's being added on to the DNA strand here in the form of plucking out these methyl groups, which actually are supposed to originally come from the food, whereas other things are going along here. Maybe these are, I'm not sure if that's supposed to be this. This is a donor of these methyl groups to this process. Now, we've heard a lot in epigenetics about the methylation of the DNA strand, but those other proteins I was telling you about, the histones, can also be methylated. But they can also be modified in all kinds of other ways. They have these other little chemical groups added to them, like acetyl groups, and acetylation and deacetylation also change the biophysical properties, the three-dimensional topography of chromatin and gene transcription in the cell. Now, why is that important? Well, it gives us a way of understanding how food comes into the body and is not just a kind of fuel for the flame, right? But comes in and is left as a trace on the surface of either the DNA strand or in the structure of the landscape of chromatin. The difference that this can make is very classically found in the agouti mouse model, which is the first model organism I encountered in watching this field. This mouse and this mouse have the same genome, the same genetic sequence. They are come from an interbred line of mice, so they're as identical twins, they like to say. But of course, the difference between one mouse and the other is that this mouse, as a agouti gene locus, is highly methylated, whereas this one is not, and this mouse is experiencing an overexpression, an ectopic bodily expression of agouti protein throughout its body, causing this kind of obesity. Now, these could be from the same litter. And so the classic experiments that these mice became iconic for showed that if pregnant mother mice were fed foods that were high in substances that would donate methyl groups, you could shift the likelihood, you could shift the incidence of coat color towards having more brown mice as offspring and less of the fat yellow mice. Or alternately, in the other direction, also with various kinds of pollutants added to the diet, you could see a shift back in the other direction. So the importance of this experiment, regardless of whether you understand methyl groups or any of those things, is that it is something coming from outside of the genome that is coming in and modulating the homeostasis, the stability of the genome over development and through inheritance. And so it's kind of ironic because this mouse was traditionally actually a model for the genetic inheritance of coat color in the beginning of the 20th century. And then in the 80s and 90s, it was a model for the genetic control of metabolic homeostasis. And it's not until the 21st century that it switches. Its body is turned inside out in a way, and it becomes a model for the metabolic regulation of genetic homeostasis. And so it's this logic of the imprint that I want to leave you with. And this imprint has a very particular temporality. Rob Waterland, the scientist who first introduced me to these ideas, shows it with this kind of slide where this period of plasticity and openness to the environment is the core paradox of epigenetic logic. It's openness to closure. It's openness to the stable modification of DNA in these early periods of life marks the epigenetic process of changing chromatin landscapes and therefore closing down these patterns of possibility later in life as the organism becomes more stable. The logic of the imprint comes very explicitly from Konrad Lorenz, the behavioral scientist who showed that if he was present when these geese hatched from the shell, they would imprint on him, they would follow him around as if he was their mother. But I also want to add that there's another temporal cycle here that is going on in these sciences, and that is the temporality of the circadian rhythm. So here's the cover of the journal Cell Metabolism, recounting the results of a study in which two groups of mice received the same amount of the same kind of food. And again, they're very, very similar. But the difference is that one group of mice has their food time restricted. So they only get it in between, I think that's probably 8 p.m. and 4 a.m. because mice are nocturnal. So they get all the food that they're going to eat in that time period and none in between 4 a.m. and 8 p.m. These mice are given the same amount of the same kind of food, but they can access it whenever. It's given ad libitum. The physiological results of eating in a time-restricted way periodically with a rhythm, that's why it's called cafe rhythm, I think, in this depiction, are put across in the differences between the two mice. And indeed, the appearance of all of the classic features of metabolic syndrome, high triglycerides, high middle body adiposity, diabetes, all of those things appear in the mice who are fed ad libitum. And they also saw a breakdown in the circadian rhythm of those mice. So this sort of sense that the imprint that food leaves on the inside is very time-dependent is, again, a break with the previous temporality. So I want to ask then, getting perhaps a little bit more distant from this kind of biochemistry and scientific result, what the metabolic politics of the epigenome are here. Here's the central dogma that we know so well from the history of genetics, that DNA makes RNA, makes proteins. We might ask, where is metabolism in this scheme? Well, generally, it's included down here at the bottom under proteins in the sense that the genes make enzymes, right? And the enzymes of intermediary metabolism run the business. So there is, in the 20th century, this sense of a hierarchy in which the genetic executive functions run the metabolic functions. But if we listen to the logics and the story of methylation, we find that metabolism is still in these kind of circles, but you might notice that this is folic acid supplement and natural folate feeding into this metabolic cycle, which is carbon-1 metabolism, and in the end, methylating substrates. Now, those substrates are, as I explained, relevant to the landscape of chromatin and the genome. The biologist Janine Lassalle has said that we should think of the genome as a kind of methylation sink. And it's not just that there's food and it's contributing stuff to methylation, but there are other things that are drawing down the supply. That is, if you have environmental toxins that can interfere with the process, that can draw away from the process of methylation. So here you have diet is the supply. But you have all these things that are sinks: inflammation, DNA repair, genome instability, environmental toxins feeding into like radiation or mutagens, causing genome instability, requiring DNA repair, viruses, inflammation, environmental toxins. These are all things that are pulling on this metabolic cycle that I showed you before. And so food and supplements are feeding in. And during development, this is very consequential, but also throughout life. So the term genome instability has flourished in the literature only since about 2004. It's a great transformation of the understanding of disease from mutation to a kind of failure of maintenance of stability. Human DNA is 70 to 80% methylated, which is very high compared to other species. And copy number variations and retrotransposons, these ancient viruses in the genome, are more highly methylated than other parts of the genome. So this process of methylation is essential to maintaining, as I said before, genetic or genomic homeostasis. So the classic definition of metabolism is the chemical processes that occur within a living organism in order to maintain life, the interconnected sequences of mostly enzyme-catalyzed chemical reactions by which a cell, tissue, organ, etc. sustains energy production and synthesizes and breaks down complex molecules. Or more simply, the definition of to metabolize is to alter within metabolism. So these things that I've been showing you, these kind of biochemical cycles, basically means that any time that a chemical group is added or taken off of something with an energetic reaction, something is being metabolized. So I like to think of the genome as being constantly, perennially metabolized. So we can come to the conclusion that nurture metabolizes nature. And this is not just my conclusion, but here is the observation of a couple of scientists writing in a book about obesity. The concepts underlying genetics and metabolism are in effect merging. There is metabolism at all levels of biological organization, from the whole organism to the cell, and finally down to the genome. Now, this matters for biochemists and scientists, but it also matters fundamentally for us. So we can think about mandatory fortification policies, the addition of folic acid, which is a methyl donor, to all enriched wheat products in the United States and many other countries around the world. We can think about the rise in shift work. I was recently looking at the list of the top 10 employers, public employers in the United States, publicly held companies in the United States. And up there, of course, are things like Walmart and McDonald's and UPS and IBM and Yum Foods, which is Taco Bell and Pizza Hut and KFC. And really being struck by this juxtaposition of the companies which run the logistics, right, of moving stuff around the country and keeping things open all the time with these fast food companies, right? And so this is an economic landscape. It's a financial and logistical landscape, but it also changes fundamentally the work and eating rhythms of all of us, right? And so I would like to think about that economic and financial landscape as a temporal, biological, metabolic landscape that we operate in. Epidemiological and medical work on people who do shift work shows a much higher incidence of obesity and breast cancer and other disorders in shift workers. Air pollution has been correlated with the undermethylation or the hypomethylation of the genome. Stress and glucocorticoids feed into neurodevelopment. Malnutrition in utero linked to metabolic disease in adulthood. As I mentioned, with this idea of the genome as a methylation sink, flame retardants, lead, pesticides, all these things which are socially and economically distributed and do not encompass bodies or imprint in bodies in socially equal ways are consequential. I could also give you a whole other talk about sugar and antibiotics, but I promise I won't. Okay, I had a whole third part, but I see that I am quite at the end, so I am going to go to the end and bring you back to this quote, right? Intermediary metabolism, the ultimate aim of biochemists, the account of intermediary metabolism, the transformations undergone by matter in passing through organisms. What I think we're seeing in the contemporary sciences is a very, very different kind of theorization. What we're seeing is the question of organisms passing through matter. And that kind of inversion helps us understand that this is a very, very different connectivity between inside and outside. I mean, things don't completely dissolve, of course, but this sort of sense that the stuff in a very temporally specific way is encountered by organisms as they move through time and space and fundamentally constitutes them in a very profound way is really, really different from the industrial, combustive, independent notion of the individual who kept the chemical laboratory on the inside of the 19th century. Now, this won't make entire sense because I had a bit about the kidney, but another conclusion is that kidneys are as interesting as philosophers, and that I hope that this series and my work contributes to the idea that there are really fascinating critical and theoretical resources to be found in the writings of the writings and the processes of the life sciences. So I'll end there with this cake topper. Eating is in my DNA, which is under the class of DNA replication humor. So thank you very much, and I look forward to your questions. Right. Okay. Is everybody up for that?
Yeah.
Okay. So the story about the kidney, the question was, could I just give a few words about the kidney? So I wanted to end with this story about the kidney. And the really, really short version of it is that in an era of really amazing, sophisticated tools by which you can fish a biological thing out of the body and and sequence it and go into a database and find out what it matches, there was a kidney biologist, a kidney specialist, working on disorders of hypertension, so high blood pressure. And the kidney is very involved in high blood pressure because it regulates the flow of blood and blood pressure and all of those things. And she found a receptor that was mutated in some kinds of kidney diseases that led to extremely high blood pressure. And when she, figured out what kind of receptor this was, went into the database, it came back saying that it was a smell receptor. And she thought she must have pulled the wrong sample from the freezer, because what would a smell receptor be doing in the kidney? And what she figured out was that it was indeed a smell receptor. That is, we call it a smell receptor because smell receptors were first found in the nose. And it was indeed this receptor that was specific to an odorant, chemical odorant ligand that came through the bloodstream to the kidney and specifically fired off the production of renin, which is highly relevant to blood pressure. What was this ligand, this specific ligand? It was a short-chain fatty acid made by bacteria in the colon. And so the bacteria in the colon, in the process of digesting complex carbohydrates that humans can't digest for themselves, would produce this short-chain fatty acid. It would cross over the wall of the colon into the bloodstream, move to the kidney, and be taken up by this receptor, and the kidney would, in response to that, adjust blood volume. Because presumably when the body is eating, it wants to circulate nutrients around more. There are these kind of reactions, these reactions of expanding blood volume and contracting blood volume in relation to eating. And so I like that story very much because it is, again, a way of thinking about the kind of categories that we set up in earlier times. We have these systems, right? We have kidneys, and they're dealt with by nephrologists, and smell, sensory systems are dealt with by other people, immune systems, metabolism, genetics. So for me, this finding of a smell receptor in the kidney was an example of this kind of strange biology that makes us think about the outside world as being fundamentally not outside, but always inside and throughout in a kind of constitutive way. So that was my story about the kidney.
Sorry. Occasionally I get to do this, but aha. So thank you, first of all. I'd like to go back, if you would, to your invocation of Elizabeth Wilson's observation that the anti-biological gestures, like the engine that starts the ignition that starts the theoretical engine, and surely that's to be explained not only in feminist and queer theory, but as you said, in cultural criticism generally, by the widespread understanding that a recourse to the biological is a recourse to fixity, right? Yes. Especially if we're talking about genetics. And so there has been, for the last couple of decades, this suspicion among humanists, when we hear the word genome, we reach for our guns, because we suspect that inequities are being naturalized, and in fact it is the very work of ideology. And what I find striking in some, not only here, but in the material you've given us for the seminar, there's a seminar tomorrow, but also so far in the series as a whole, is the redescription of the genetic as, in fact, the realm of flux. And so, especially with regard to the methylated DNA, your work also, you're following with the on chromatin, it seemed to me in one of the things you circulated to us that the argument eventually contests the very distinction between epigenetics and genetics. And, okay, could you say more about that and where that would take us?
Sure. So people...
Small question.
Yeah, small question. Now, the sort of basic definition of epigenetics that you'll get if you look it up, or if you ask a biologist Or if you write an article and you send it to an editor and they write back and they say you have to put this definition of epigenetics in your article, right, is that it is epigenetic. It is the processes above the genome that change gene expression across generations, either of cells or more controversially of individuals, without changes in the underlying DNA sequence. So the very definition of it depends on sequestering the genetic to one form of change and the epigenetic to this kind of buffer layer above, right? But in the sciences of something like genome stability, what people are working on is this sense that if you have this crazy human genome that's almost 50% repetitive sequences, which is very dismaying, I think, because it's a little bit undignified to be riddled with repetition, right? Why do we have so many repetitive sequences? These repetitive sequences, it means that if I had, A, T, T, C, G, I would have that sequence exactly, A, T, T, C, G, repeated 100 or 1,000 times in a little stretch. And this can either be in non-coding DNA or in genes. So a classic example of copy number variation or repetition is the gene for amylase, which breaks down carbohydrate in the spit. I might have 9 copies of the amylase gene, and you might have 15, and you might have 50, right? And that would correspond to differing levels of amylase in our spit. That's a harmless kind of copy number variation. But it can have really profound consequences also for neurodevelopment, especially if there's a bit missing or a bit duplicated that's really consequential. So what maintains the stability of DNA copying when you have all of these repeated sequences, and they don't line up so well when things have to line up in the cell, or some of those repeated sequences are actually still active retrotransposons that can excise themselves from one spot and put themselves into another spot. Well, it seems like organisms maintain a kind of handle on that difficulty by really heavily methylating those parts of the genome. And this is the same also for the ends, the telomeres on the chromosomes. Those are heterochromatic. They're very, very heavily methylated and actively maintained as closed, right? And so this is one of those places where you can say, well, the epigenetic bit is all the mechanisms of methylating and the genetic bit is the mutation. But actually, the physical stability from cellular generation to cellular generation, or perhaps from individual generation to individual generation, is the maintenance of stability through epigenetic mechanisms. So there is this kind of partitioning of those things, but that's very artificial. And that's why I like to talk about chromatin and rather than DNA methylation by itself, because you only ever get DNA and DNA methylation as an artifact of chemical separation. DNA doesn't come by itself. It's always chromatin in the cell. And so I think that fundamentally the separation between epigenetics and genetics has been very productive for scientists, and also in certain ways of self-definition of a style of scientist, or a novelty, a departure from classical genetics. But it's not that clear to me that they're that separate. And there are many ways in which the kind of stability of the genome is linked to the constant maintenance of it as a physical object. And, we're still recovering from DNA as information, right? It's almost anti-intuitive to think about it as this temporal, dynamic, ever-changing object. I saw a scientist giving a presentation in which he showed a film. It was some crazy high-tech form of imaging, single single molecule imaging, where they had managed to tag something onto a gene as it was being expressed. And they could show the gene itself moving in the nucleus as it was being expressed. And everybody in the audience was just flabbergasted by it, because they were so used to seeing these sort of jelly bean descriptions of the DNA as a kind of railroad track and the polymerase moving along it. And, and someone next to me said, who would have thought that genes move? So I think this kind of temporal dynamic body is the genome and the epigenome at the same time.
Oh, go ahead. Oh, sorry. Okay.
I don't even think I need this, but I guess this rides back off of that question, which is I'm imagining all sorts of interesting challenges of mentoring students in the social sciences, so I'll ask about that half, because I think of this talk model, this signal model, as being very dominant, sort of the way people want to think about maybe even something so... The first thing that came to mind is digestion, and like how your approach would transform the ways we even talk about, like digestion is my body talking to me, my body telling me certain things are not good for me. But maybe some of the big mystery of digestion that I see on a lot of like forum boards or whatever is that people suspect this is an inadequate model. So like, what is it sociologically, and that you encourage your students to maybe pick up on, maybe clues in the way people talk about the inadequacy of talk as a model for understanding the body's communication, rather than this other version, which is a co-constitutive understanding of this transformation, right, in progress. It's a very different paradigm. And so I just wonder how you mentor kind of students into picking up on that and developing new approaches to research and so on that work in the social sciences.
Thank you for the question. The first thing, I guess, that I try to foster is a certain, if not bilingualism, at least comfort with more than one form of language or thinking. And that comfort, often in terms of social science students reading biology or reading science, also means listening to skepticism, or listening often to the skepticism or the debates that are happening within the sciences themselves, because there is so much auto-criticism within the sciences. So I tell them to go and sit in on a journal club meeting and listen to people argue, or if they're interviewing people, find some young people who are actively searching out the cracks in their mentors' assumptions so that they can find their own ground of self-establishment. So there are actually a lot of resources within science itself for having this kind of thinking about the structure of assumptions and those kinds of things. But you're absolutely right also that I think that one of the reasons why we see this shift and renewal in metabolic sciences is that the old explanations simply did not work. And just a few days ago, I was giving a talk at Columbia where I was paired with a diabetes specialist, and he got up there and he was talking about energy balance, calories in and activity and those kinds of things. But then he kept using himself as an example of not being able to, even though he knows that, of not being able to make his weight shift, right? And so I think the kind of fundamental manifest inadequacy of many of these explanations, which we really have inherited from a long time ago, food as fuel and as building blocks, it's really hard to think about those as having been enormously productive and still partially in action and partially true within our structures of being true, but just not working either as therapeutic explanations or as self-explanations for people. So I do think that listening to people self-experimenting is an enormous resource for students. But I also, also will just say it's really hard. It's really hard to get economic sociologists or people used to a very certain kind of explanation to ask what's the biology of that, or where might biology or biological thinking enter into something that you have always only thought of as social. I would add also that history is a really great resource for rethinking that, because you can go back to even a classic like Durkheim, right? And you can see the ways in which disciplinary specialization and the walling off of some territories from other territories was essential to the establishment of the discipline in the first place. And so when you can dissect that through a kind of close reading, then it does denaturalize these things a little bit.
I have a million questions I'm not going to ask right now, but this
All
was
right.
very fun to hear. And I'm way shorter than this. But so thinking about that, the other part of that question really is to ask, okay, so you've got your students in a social class, for example, or economic sociology, or in my case, I have the students who are working in a class on gender and science reproduction, where we taught, we read your book, and we're working with members of a lab over in the College of Agriculture who are studying, they're studying health effects that can be moved from animals to people. So it's a lab. And we would meet together. And the question there is, how do you make it possible for the people on that side to engage and move across the boundary and have both the time and the linguistic resources and the patience and the et cetera to talk to us? Because in some ways that's the part that's, if I make myself clear, that's the hardest part, is to get it really a two-way exchange. So, is this a problem you've had? Or are you mostly talking to sociologists or people in the,
Well, no, I spend 50% of my time in a department that is housed in the Division of Life Sciences, right? So the Institute for Society and Genetics is the equivalent of a department, because we house faculty FTE and we host an undergraduate major and minor in the Division of Life Sciences. And so I guess the facile answer is that your people need to take our major, human biology and society, because they can earn a Bachelor of Science degree in which they do all of the regular things that, say, a pre-med would want to do, biochemistry and all of that stuff. But they also choose another focus, and that can be whatever they want. It can be philosophy, or it can be history, or whatever they want. But they all have to take this backbone, this integrative core, in which they take classes that are called things like ways of knowing. And you just simply go through and you say, what is it to know something in this discipline versus this discipline? What's the difference? And where does the distinction come from between quantitative and qualitative reasoning? What constitutes proof? Why do some people not believe other people? And so and so when you equip people with the tools, not necessarily of how a molecule works, but the tools to understand the framings of different kinds of knowledge production and give them equal legitimacy and footing, then they become much more flexible. They become much more able to think about things as not always having to fit perfectly together, like you have humanities pieces of the puzzle, and you have social science pieces of the puzzle, and you have science pieces of the puzzle, and you put it all together and you get this lovely picture. Actually, you have 3 different puzzles. Yeah. And so having some realism about that and realizing both what incommensurability is and what commensurability is, I think that's something that I worked very hard pedagogically to do. I don't think it's very easy at all. But, I also do have perhaps unwarranted optimism about it, because I feel very strongly that that many of the kind of looming enormous cultural and social problems that we face socially are necessitating and also inspiring students to think more than one way in that way. So, you know, I've been obsessed with antibiotic resistance ever since I started teaching about it. I didn't know anything about it. I picked it as the kind of example problem to start teaching the introduction to the major with, and I myself got completely caught up in the problem. And I think that it is absolutely possible to train students to be a little more flexible. It's very hard when they're being formed professionally to take them into one class or one whatever. And, you know, I've been in many, many interdisciplinary settings where juxtaposition is expected to produce togetherness. And I really think that understanding how a set of people from different disciplines can share a concern, and that it can have some overlapping parts, but not everybody has to be working on the same thing, right? Just more the question of how each can be enriched by the shared overlapping parts of the problem is part of that. Then there are power differentials on campuses and all those things, but we probably don't need to rehearse those.
Okay, this definitely wasn't going to work for me out there. So I don't know if this question is going to make sense, but you know that slide that you had in which you were talking about how metabolisms get thrown to the bottom of thinking about the genomic sequencing. And so what I'm trying to figure out is I can definitely understand from a public health perspective what the productive erosion of the barrier between genetics and epigenetics might mean. Okay, that I totally get. My question is, what does that mean when you're working on therapy? So the classic one I'm thinking of is reverse transcriptase inhibitors. And would it change how we think in terms of the fact that that does depend to some degree on the original stability model? But there might be other, HIV therapies that wouldn't depend upon reverse transcriptase inhibitors. Which does that make sense as a question?
Yes.
Thank you.
Yes. I have to think about the answer for a second, though. So one mode of answer is pretty general, and that is to say that along with the hierarchy of genetic things running metabolic things has been the assumption that you had to work on the gene or the signaling cascade, like that was the place of therapeutic action, right? And so all of this focus on mutations or oncogenes in cancer, for example, that lead to cell growth. And it was always assumed that you saw different metabolism in tumors long downstream from that you get these mutations and then you get malignancy and then, you get these tumors with disordered metabolism. But in the field of cancer metabolism, there's a huge renaissance in the area of cancer metabolism. And the idea being that there are many ways that you can think really differently if the disordered metabolism comes first. And that many of the enzymes that were classically thought only to reside in the cytoplasm, doing things like turning, excess sugars into fats as storage, they're actually active in the nucleus. And what are they doing in the nucleus? Well, they're making things that are essential cofactors and intermediaries in chromatin acetylation and things like that. And so we've always thought that signal transduction runs metabolism, but there may be many other processes, and it's not either or, it can be both, right? Many other processes whereby the inappropriate shutting down or opening up of various things precedes the mutational events or these other things. And so there's very active work in the pharmaceutical industry to find these molecules like HDAC inhibitors. So HDAC is an enzyme that modifies chromatin. And so the strategy is actually really different.
I was going to say the strategy must be very different than that.
Yes, yes. And in the case of reverse transcriptase inhibitors, you're referring to HIV therapy, right? So there I might give...
That's just a model that I know.
Right, right. But I might give a slightly different answer there, which is more about the questions that have been so far unthought. So there are many aspects of HIV that have been treated as kind of side effects. So, for example, HIV in children causes accelerated aging, and many other metabolic side effects, right? And there may be many ways in which nutritional strategies for treating HIV have been seen as quackery, or nutritional strategies for treating autism, for example, have been as a better example of being treated as quackery. But I'm sure a lot of it is quackery, right? Anybody who tries to sell you a therapy that says this is the answer is probably wrong. But I would say that many of those things are either underthought or under-investigated exactly because of the hierarchy put on things that are very respectable, like intervening in DNA transcription and RNA transcription mechanisms, and those things which are unreputable because they're just nutrition science, right? And so there I would say there's probably not so much a question of the new cancer remedies versus the old cancer remedies as a domain in which there are many roads not taken exactly because of the very strong hierarchy there.
Thank you.
Thanks again. We have a little reception afterwards, so if everyone's invited, right next door. And thank you.
Thank you.